Hello guys, today we are going to study about class 12 chapter biomolecules. Let's see the definition of biomolecules first. So biomolecules are macromolecules which are naturally occurring in biological system. Examples of biomolecules are carbohydrate, proteins, nucleic acid and vitamins, etc. Okay, and in this video we are going to study carbohydrates. So let's see the definition of carbohydrates. So carbohydrates are polyhydroxyaldehydes or ketone or compounds which give rise to such units on hydrolysis. Means they contain hydroxy as a group, functional group OH. Apart from that, aldehyde CHO. So either they contain CHO and OH as functional group, or it could also contain ketone C double bond O R C double bond O R and hydroxy OH as functional group. Then classification of carbohydrates. So carbohydrates are classified into three broad groups in accordance with their behavior on hydrolysis as monosaccharide, oligosaccharide and polysaccharide. So these are the three classes of carbohydrates: mono, oligo and polysaccharides. So what are monosaccharides? These are carbohydrates which do not hydrolyze further into smaller units. So we cannot hydrolyze them further. Hydrolysis is addition of water. So by addition of water, we cannot break them further. So those are monosaccharides. Examples are glucose, fructose, ribose, etc. Next is oligosaccharides. So these are the carbohydrates which on hydrolysis give 2 to 10 units of monosaccharides. For example, sucrose is a disaccharide. If we break sucrose, we'll get glucose and fructose. Okay, by hydrolysis only. Similarly, maltose is obtained. It is also disaccharide. It is obtained from two glucose molecules. So when hydrolysis of maltose took place, two glucose molecules are obtained. Okay, so it could be between 2 to 10 units. There are trisaccharides also, tetrasaccharide, pentasaccharide and so on. Next is polysaccharide. These are the carbohydrates which produce a large number of monosaccharide units on hydrolysis. Large number means they contain more than 10 units of monosaccharides. Example is starch, cellulose, etc. So these are the three classification of carbohydrates. Then next part is reducing sugars and non-reducing sugars. So those carbohydrates which contain free aldehydic or ketonic group and reduce Fehling's solution and Tollens reagent are called reducing sugars. So in this all monosaccharides are there. Okay, all monosaccharides are reducing sugars. Okay, they reduce Fehling's solution and Tollens reagent. Therefore, they are called reducing sugars. On the other hand, non-reducing sugars, those sugars which do not have free aldehydic or ketonic group and do not reduce Fehling solution or Tollens reagent are called non-reducing sugars. Example of that is sucrose. After that, we'll see now preparation of glucose. So two preparations are there. First is from sucrose. Sucrose is C12H22O11, and when we perform hydrolysis of sucrose in presence of acid catalyst, so this H plus is acid catalyst. It could be obtained from HCl or H2SO4, and hydrolysis is addition of water. So when hydrolysis of sucrose takes place, it breaks into glucose and fructose. Both are having same molecular formula, but they are having different structure. So molecular formula of them is C6H12O6. So that's the first preparation of glucose. And second is from starch, that is a polysaccharide. Commercially, glucose is obtained by hydrolysis of starch by boiling with dilute sulfuric acid at 393 Kelvin under pressure. So starch formula is given C6H10O5 whole bracket N plus N number of H2O. In the presence of acid catalyst at 393 Kelvin and 223 atmosphere, it gives N-glucose. Now we are going to discuss the structure of glucose and fructose. In earlier preparation, we saw both are having same molecular formula C6H12O6. But see the structure now. How to draw structure of glucose first? So draw 6 carbons. We can count 1, 2, 3, 4, 5, 6. So 6 carbons. Draw them first. Then first carbon is aldehydic carbon CHO. Last carbon is CH2OH. And on the third carbon, write H on the right side and OH on the left side, and a complementary right on the other side. Okay, so CHO at first carbon, CH2OH at last, H is at right side, and for other carbons OH is at right side, and you have to write complementary of that on the left side. So that's how we can draw structure of glucose. So glucose contains aldehydic group as functional group along with hydroxyl group. Okay, total 5 hydroxyl group and 1 aldehydic group is present in glucose. And in case of fructose, we can write like this. Draw 6 carbons. First carbon is primary alcohol CH2OH. Last is also same CH2OH. Second carbon is ketonic carbon C double bond O. And third carbon similar to glucose. Hydrogen is on right side and remaining are having OH on right side, and then we'll write complementary to the left side. So that's how we can draw structure of fructose. So that's how from this diagrams it is easier to distinguish between glucose and fructose. Glucose contains aldehydic group and fructose contains ketonic group. Now next part is the structure of glucose. So how glucose structure is determined, that we are going to see here. So glucose is a 6 carbon straight chain aldose which has one aldehydic group CHO, one primary hydroxyl group CH2OH at the last carbon, and 4 secondary hydroxyl group CHOH. If the OH group attached to C5 is on the right side, the glucose is assigned as D configuration. If the OH group attached to C5 is on the left side, then it is assigned L configuration. Plus minus signs represent optical rotation as dextro and levorotatory respectively, and there is no relationship between D and L configuration. So if we see structure of this glucose, the this carbon C5OH is at right side. So in glucose, how many chiral carbons are there? There are total 4 chiral carbons in glucose. Carbon attached to 4 different groups, and out of them this one is the last chiral carbon where OH is attached to right. Therefore this glucose we can say it is a D glucose because last chiral carbon's OH is at right. Therefore it is called D glucose. If it is at left, then it is known as L glucose. Like that we can name it. And there is no relationship between dextrorotatory and D and levorotatory and L. That dextrorotatory and levorotatory optical rotation can be obtained by using instrument called polarimeter. Next is a molecular formula of glucose was found to be C6H12O6 on the basis of its elemental composition and qualitative property which you studied in solutions chapter. Second point, the 6 carbons in glucose molecule form a straight chain. This was inferred from the following observation. Glucose gives N-hexane on prolonged heating with HI. So how to prove glucose is having 6 carbons? So if we heat glucose, CHO, CHOH, 4 secondary alcohol and CH2OH. If we heat glucose prolonged heating with HI, it will give N-hexane. And from that we